we get experimental. SciAnts. We're curious, non-judgmental.
A smidge of something scary. I think y'all can do it. This is actually a creature that has spread throughout the south of the United States and is quite invasive. So here's the title of the paper. It is called Sea Lamprey Enlightens the Origin of the Coupling Retinoic Acid Signaling in the Vertebrate Hindbrain Segmentation. Good Lord, that is a mouthful. Basically what they're doing in this paper is they're looking at brain development, and they're finding that there's similar molecular and in terms of molecules that are used like retinoic acid, as well as the genetics of brain development, especially in the hindbrain in both mammals like humans as well as the lamprey. This study was done at the Stowers Institute, so this is not a direct research lab per se, but rather it is just a large institute. They're not tied to any university. There's no need for teaching. The lead researchers, quote, they're saying that they study the hindbrain. It's the part of the brain that controls vital function like blood pressure and heart rate. So basic functions of an organism to survive. It's looking into like the evolution of complexity is what they're trying to look at. And they find that these sea lampreys, like other vertebrates, they have a backbone and a skeleton, but they're missing a jaw. But yeah, so the evolutionary origin, GK, between jawed animals and jawless animals, that split is 500 million years ago. So the fact that we're looking at an animal that's 500 million years distant from us, at minimum, and it has similar brain anatomy and brain development is pretty remarkable. So they wanted to think the whole goal of this study was how the vertebrate brain evolved, and if there's something unique, the jawed vertebrates that's missing in the jawless, right? So do we have something that the lamprey doesn't have, and vice versa? Why does this continue in the jaws? Oh, gotcha. Well, so you mean like these animals? They have an ecological niche, but the way they survive is perfect for them where they don't need the jaws. And so I think it's a right way of putting it in quotes, right, GK, of the idea of better, because better for us isn't necessarily better for anyone else. Like other organisms might be perfectly adapted and suited in a very different state. This is the same issue when you ask if an insect or any animal is smart. Right? Because, they might not have like intelligence like us, but they might be smart enough for whatever they need of how they're living. So that's, I think, the same thing. It's not necessarily better. Well, it's better for us that we have jaws, right? But for them, they are able to survive in a particular ecological niche without it. But yeah, they were looking for the genes and like in previous studies to figure out how the brain gets oriented the right way. And so in this paper, they found that there is a signal that goes from head to tail that acts as a gradient. So high concentration in the head, low concentration in the tail, and it signals the neurons to connect. And the Q is actually retinoic acid or vitamin A. And that vitamin A is the same Q that's in mammals as well that helps with that brain patterning. And so it's really cool to see that same patterning and that same signal occurring in two very different levels of complexity, right? And so the thought had been is that because there's no jaw for these lampreys, that they don't have the same kind of hindbrain as you and I do, because we always suspect like the hindbrain has to do with much more like talking and this kind of information perception and eating and like mandibular movement. And so why would the animal have that? See, it looks like the lamprey does in fact have that hindbrain. It's just the formation of it is a little bit different. Started by the retinoic acid the same way as in us, and there's maybe a little bit of different segmentations of it, but it's still the hindbrain. And so yeah, this was just a study in looking at common ancestors. We'll look a little bit more at the paper itself. I really liked it because they had a bunch of different techniques. They had CRISPR that they did in the lampreys. They also did genetic manipulation. They did manipulation via small molecules to really prove this point in terms of brain development. So they really went above and beyond to really show and highlight all the different ways that the brain is acting and functions in this kind of scenario. So I was like, hats off to them for doing a really, really good job on what could have been an extremely complicated study. It does, and actually, Ingers is right. So you can see it on sharks a lot, and you can even tell when there's been an injury to the animal because of this, because it's suction cups onto the site, and then it's like, imagine like a plunger removing, and then you just have like this circular, like, of like the meat that comes out with it, because it's like a little circular saw. No jaw though. So yeah, they looked at brain development in these animals, and they found that there are similar genes that are governing the development of their hindbrain. So they did find the genes that govern this hindbrain development in the animals. They are still parasitic, Ulo, like where they belong in the ocean, but those animals have co-evolved with them to survive, so that the death rate is not as high. There's still some because it's a parasite and sometimes they overbore, but most of the animals survive. And there's actually resource competition between the two animals, so if they're coming in, you know, and parasitizing, whether they survive or not. So no, Gurs, the point of this paper was they were looking at hindbrain development. They found that there is retinoic acid is a signaling molecule for the development of this brain, and they also identified two genes, the CYP26 and the ALDH1A genes, that are involved in the development of the hindbrain of these animals. And I thought it was really, really cool. Yeah, so that there was no difference, at least for these animals, Gurs, in salt and fresh water. I say with that caveat because I don't know. There may be, in some instance, right, a difference. There didn't seem to be anything here. They also didn't go into looking at the differences in water, so take that with a grain of salt. But they were actually able to turn off these genes using a couple of different methods. So the way they did was actually quite remarkable. They used both drugs and genetics to turn off these genes. Like, so the drugs were inhibitors of these genes that they had already identified before. And then they also went in and did genetic engineering on these lampreys to turn off these two genes. So I just really liked how it was done in a multiple different avenues of research. So Gurs, I don't actually know where the drugs came from. See, they did a sequencing experiment to figure out what genes and how they turned on and off. I mean, here they're looking at the development of these genes. Let me actually see if there's anything that we have based on the drug here. Yeah, this Terazol. So it looks like the drugs, we actually know the mechanism of action, so it must not have been a new drug, right? This must have been an existing drug that they already had, just off the shelf. They didn't go into like where the drug is from. I mean, it's actually a smart way of doing it again. So like even the explanation of what the drug does. So you go from like this palarazole, right? The inhibitor of the CYP26 enzyme, which is the enzyme they identified through the RNA sequencing that increases levels of retinoic acid. And so that's already the mechanism by which that they're working. So this drug inhibits the gene candidate that they found from the RNA sequencing that messes with the levels of retinoic acid. They then also looked at a second drug, this DEAB, that inhibits the ALDH1A2 enzyme that also changes levels of retinoic acid. So Gerd, I was just looking and seeing they didn't make the drugs for this study, they already existed. And like you were saying, it's kind of like, why does that already there? You know, like why, I don't know the history of these two drugs. So that plate, 500 drugs that have never been before tested. And the idea is use those drugs and test for a phenotype that you might be interested in. And just see what you might end up finding. And there's no way of knowing. You might find out something brand new. You might find something very weird. And I think that's a really interesting way of thinking about it, you know? Oh, for sure. But Gers, I would argue it's even scarier, right? Because if you have a whole plate of drugs, you're like, I have no idea what any of these things do. Let's check it out. And it's like, you're only testing for one particular question, right? Of like, does it affect this biological process that I'm asking a question about? Versus, you know, how much it could do. But yeah, so these two drugs that they had were really cool in that they were able to help like confirm what they hypothesized about the lamprey. They also had some really nice imaging in the development of the brains of these animals as they shut down certain genes and turned on other genes. And then their final thing that they just kind of snuck in here is they had the drugs, and yeah, to further investigate, they were like, oh, we made like these lampreys that light up if you turn off things or not. And then they did, let me find it, CRISPR-mediated mutagenesis of the genes that they're interested in. So they went in and actually mutated the genes that they were targeting. So this is why I really like this paper, right? It's the beginning point is like, we think these two genes are involved, the retinoic acid mediated and hindbrain formation of a lamprey. Let me show you 10 different ways that why we think this is the answer, right? So some people could have just stopped with the drugs and be like, look, it's targeting this and we're done. or maybe the transgenic. So the glowing one, Gers, is we're going to make the animal's brain glow whenever there's an expression of this gene, right? And so it's like, oh, look, this gene that we think is important is on in the brain at this time, therefore it must be it. Done. Or they did RNA sequencing too, right? And they found that these genes are really highly turned on in the hindbrain. But instead of just having one of those, they went through and tested in so many different ways that it's almost beyond a shadow of a doubt that, you know, what they're saying is probably correct. Yeah, I think it's very solid. They have a lot of experiments. They even have, like, models and how they think it works, like the gene levels and connection to the retinoic acid signaling. It's a lot of nitty-gritty, but I think it's a really nicely done paper. And they, again, more models and how they work, how they suspect it works. And I just thought it was cool because it's a non-model organism, like it's a lamprey, right? You never hear about a paper on a lamprey because no one works on lampreys because they are difficult to work with. And yet, someone worked on a lamprey and got the experiments to go, and it's really cool. So it's just cool, again, to think about the biology of this animal, but knowing that they were not studied in this paper for biocontrol or their survival in salt or freshwater, like Gers was mentioning. They were studied for brain development. So it's an example of studying an animal, not just for the sake of studying it, but it's the best animal to study for your question. And if you're looking at the evolution of brain development, it was the ideal point in time to study it. The host number timing, Claire, is difficult because they're invasive. So even though, like in this video, like, oh, they don't actually kill the fish, that's actually an out-of-date metric. It does turn out that mostly the host will die because they're not adapted for this. They're coming from the ocean, right? And so their hosts are like, this is weird, I don't know what's going on. They actually end up dying. So there's a huge number of fish that lose their life from these lampreys, and so they can stick on longer because there's not a natural defense for the host versus, you know, what like would be elsewhere, like in the oceans, right, where there is a natural defense there. I guess it would be hard because you'd probably have to double tag the fish and the parasite in the water. Yeah, that probably also affects it too, like skin thickness and how easy it is to take on. And probably with the sharks, because they're adapted, it's not as likely for them to have a long-term negative or as negative of an effect, right? Because there's competition. So maybe that also influences that they can stay on longer because they're not gonna like be destroyed by these. Shark age, I think that's the case. Let me check on that for you, Gers. 450 million years. So yeah, they've been mutual for a long time.